want to discuss a little bit about what it means to use the DNA. So, think about your eyes. You've got retinal cells, they have rods and cones, there's proteins in there that hold certain molecules that are photoreceptive. And so you need these molecules and these proteins to be expressed in those cells so that you can see. You don't need your fingernails to do that. The cuticle just needs to grow keratin and link it together and make hard versions of what your hair is, just cross-linked and stuck together. So these do very different things, and yet they both have a full copy of your DNA. Whenever you have a cell, it's a nucleus with some DNA in it, before that cell can split, it actually has to duplicate up your DNA. And so it has to have 2 full copies of your DNA. It kind of spreads them off on either side, and then it divides down the middle, squishes in, and you get 2 separate cells. So every cell in your body started from the first egg cell, and so every cell in your body has a copy of your DNA, all of your DNA. So why don't we grow fingernails in our eyeballs? Why don't our fingernails photorecept? What's going on? For this, there are different ways to turn on, or more importantly, turn off DNA. Large sections of our DNA doesn't code for anything. So the sections that get turned into proteins are called genes. A gene codes for a protein. But usually you'll have a long section of junk DNA, and then there'll be a chunk that is a gene, and there'll be a long section of whatever, and then there might be another gene here, and then a long section. And sometimes these can be hundreds of thousands of separations. Like, there's a lot of our DNA that is not turned into anything. Now, we used to not know why it was there. We thought it was just random noise. Recent studies have shown that that's actually important for transcribing. We need that space to keep the genes a little far away so that changes, damage in one doesn't hurt another. And also when we try to copy one to turn into RNA, we don't accidentally start copying the other one. That spacing gives us some flex to work with and keeps each isolated. And so it's kind of useful. Well, still, there's plenty of genes that we just don't want. So how do you get that gene to turn off? The first one here is our photoreceptor. Some photoreceptor gene, and that cell doesn't need it. We need that gene turned off. Modern study has brought us what we call epigenetics. Epigenetics literally means above the genome. This is the on and off, so which genes are on, off. Now, this is unique kind of to each cell. What'll happen, long story short, anywhere along the DNA where there are cytosines, they'll slap a methyl. So Me, the CH3 gets attached to it. If you have a bunch of cytosines in your DNA, it's supposed to be double helix, sorry, and they all start getting methylated, this acts as a signal. There's other proteins that crawl across your DNA. If they see a bunch of methylated cytosine, they turn off that gene. The DNA itself is not actually one long strand, so your double helix, I mean, it's a giant string. If you ever wadded up a piece of string, it turned into knots. You can't really put that in the cell. It would just tangle itself. And so we actually have these very large proteins called histones, whose only job is to have the double helixes wrap around it in order to spin up like a spindle of thread. Think of a little bobbin of thread with thread wrapped around the spool. That's what the histones do. there'll be a series of histones, and so the DNA will just wrap around it. I guess I'm doing a vertical view. If you do a horizontal view, here's my histone. The DNA comes in, goes around, goes around, and then comes out this side. It fits into a groove in these large proteins that I'm not doing a good job of drawing. But the DNA fits in that groove, and so it just wraps around the histone. And then you have these large series of histones that group together. These make up chromatin. Chromatin, chromosomes, if you think of the name. Chromatin's large collection of histones, and a large collection of chromatin is chromosomes. And so you can make these looser, or you can make them tighter. They can have multiple histones very close, and if that's the case, you get closed chromatin. So closed chromatin, close histones, turn off gene. Effectively, they pull in too tight, and you can't unravel the DNA. You can't open it up to read it. Where open chromatin is spaced out histones. These are active genes. These genes can be read. They can be activated, they can be copied, they can be turned in RNA, sent out to the ribosomes, and the ribosomes can make the protein. And so there are large sections of our DNA that don't code for anything, but even the sections that do code, some of them are just deactivated by twisting them up too tight to be read. So this is how our different cells do different things. A muscle cell needs to grow the parts to contract. A brain cell needs to make all little dendrins and reach out to all the other neurons. Your eye cells, your liver cells, your white blood cells, they all need to do different things, and this is how they do it. They literally build different combinations of proteins. They make some that none others do, and they don't make some that many others do. Just depends on what the cell needs to do. And just as an example, it can get a lot more complicated than that. We are eukaryotes. Eukaryotes do funny things, like you can have a chunk of DNA, and then, you could be trying to read this bit. And so there could be initiation, the start code that tells the transcriptase, hey, read this, here's a gene. And then way over here is termination. And so you would think I would read from the start to the end, except no, we just have bits in the middle that we call introns. And the intron says, yeah, cut me out. You don't want me later. Copy me, and then get rid of me at a later point. And so you would copy to here. Well, you do the whole thing, and you would cut out this middle bit, and you'd make a gene that was smaller in the end. But we just didn't get rid of that part in the middle originally. Like, it was there, we just had some part signals that had us remove it. And so you end up linking together these other parts that aren't the introns to make your final gene. And so this is something eukaryotes tend to do. We are still a little bit puzzled by it. We just know that they do it. So things that we are researching, and we'll discover more about in the future. A few other neat things this DNA can do. If you've got, I'll simplify it, 1, 2, 3, 4, 5, 6, 7, 8, 9. So you have 9 base pairs. Normally, we read every 3. And so this would give you, translate into 1 amino acid strain. Well, if you mess up slightly and started reading from position 2, you would get a different combination, assuming this went on for a while. Whatever your ATC, GGG, whatever that combo is, by moving it over one, you get a different 3-letter pairings. And if you were to accidentally do this a little bit more, 5. You would get a third set of possible combinations. One combination goes, hey, 1, 2, 3, I need my matching pair. Another combination, however, says, no, I'm 2, 3, 4, I need my matching pair. And the other one says, hey, I'm 3, 4, 5, I need my matching pair. Each of these are different 3-letter codes. Right? So, like I said, you could be AA, well, that's too many A's. Let's say this was A, T, G, C, G. Right? Well, you're normally here where you get an A, T, G. But if you're offset by 1, it will look like T, G, C. And if you're offset by 2, it's G, C, G. These are very different codes. If you accidentally shift over by 1, you end up producing an entirely different protein in the end. This is usually just a bad protein, and on the rare occasion this happens, nothing happens, we get rid of it. Generally, this is a mistake, but it doesn't have to be. Overprinting is the term for it, and it can happen. This is actually common in viral DNA. So you don't want a lot of DNA in your virus. It needs to be small, it needs very little to reproduce quick. And so if you need 3 separate genes as part of that virus, so you make 3 different proteins, you either have to have a really long piece of DNA and that has to get read, or you can have one piece that's a little flexible, and if you pick the combinations just right, you can get 3 separate genes out of it. Your first protein will look one way. Your second protein, because it's shifted over, might look very different. And your third one, because of its combinations of letters, might do something very different than the rest of them. Because the amino acids that you would order would be fundamentally different amino acids, you would get a very different molecule out of it. It's rare still, but it is actually kind of neat that in viruses, just because of how fast they reproduce, selection has actually made this reasonably possible. Generally, for organisms like us, this is not a common thing. Oh, I'll take it back. Human mitochondrial DNA also does this. So that kind of technically is a bacterium. The mitochondria they're part of us now, but the DNA contained in mitochondria is thought to be mostly leftover from when it was a bacterium way back in the evolutionary past. But it does technically have a little bit of overprinting. So we do this in our mitochondria. Viruses are the most common sort. And there you go. So in terms of DNA, I wanted to highlight a little bit of how it actually gets used. And so instead of just the physical structure, what are some of the ways that it plays out? How do we turn it on and off? Epigenetics itself is a very important emerging medical testing field, because we can start to say, hey, yeah, a good chunk of your liver cells seem to not have this dehydrogenase in them anymore, so you need to stop eating blank or blank, or you need to stop drinking this or that. We might actually be able to personally assess what that person's health risks are, instead of the general health risks of people at their age and lifestyle. Some people just, their livers hold out, others they don't. And we'd be able to start looking and seeing what's on and off, what took damage, what didn't. Because some of these genes get turned off randomly. If you get exposed to chemicals that can transfer methyls, which there's a lot of those, our body kind of does it naturally on its own. But if you get a lot of methylating chemicals, they can occasionally accidentally methylate DNA. And if that happens, it's possible to turn off genes in some cells that they weren't supposed to be turned off in. And so, decreased function as you age, part of that is thought to be accidentally disabling some of the genes necessary in various cells. And so we might be able to see that, and even one day, maybe, turn them back on. But we haven't quite gotten there yet. We know you can unmethylate the DNA. We don't know how it's targeted and works. But there's definitely times that your genes get turned off for a while and then turned back on later.